Ladies and gentlemen, my name is Adam Bownik, and I would like to present you the results of our latest experiments on the influence of cyanobacterial metabolites on behavior and biochemical parameters of rotifer Brachionus calyciflorus and Daphnia magna. The study was performed in the cooperation with Professor Małgorzata Adamczuk and Professor Barbara Pawlik-Skowrońska at the Department of Hydrobiology and Environmental Protection of the Faculty of Environmental Biology at the University of Life Sciences in Lublin, in Poland. Cyanobacteria may produce a variety of toxic agents such as oligopeptides, including microcystins, anabenopeptins, nodularin, or alkaloids such as anotoxin A or cylindrospermopsin. These metabolites were documented to induce different effects, toxic effects, in aquatic invertebrates, fish, or terrestrial animals such as dogs. It is to note that in natural scenario, many variants of oligopeptides and other cyanobacterial products are released by cyanobacteria at the same time, and aquatic animals are exposed to mixtures of various oligopeptides, which may induce different antagonistic, additive, or synergistic effects. Little is known on the effects of cyanobacterial metabolites on swimming behavior of aquatic invertebrates and different physiological or biochemical effects and mechanisms that mediate these effects. Therefore, the aim of our study was to assess the influence of single oligopeptides, microglycan-FR, anabenopeptin A, and microcystin LR on the swimming behavior of Brachionus calyciflorus, and to elucidate possible mechanisms of the toxic action by analysis of the influence on the nervous system by determination of catecholamine neurotransmitter activity and by determination of the influence of muscle structure. We here analyzed the F-actin activity in the muscle structure. The rotifers were exposed for 24 hours to single oligopeptides, their binary and ternary mixtures. Swimming speed was recorded with a digital camera, and the video clips were analyzed with tracker computer software. Catecholamine neurotransmitter activity was determined by staining the rotifers with ES517 probe and analysis with fluorescent microscopy. Muscular F-actin structure was determined by staining the rotifers with blue phalloidin dye and analysis with fluorescent microscopy. The type of interactive toxicity of the binary mixtures was determined by calculation of effective concentration value and isobolic analysis. This type of analysis shows whether oligopeptides in the binary mixtures induce antagonistic, additive, or synergistic effects. The type of interactive toxicity of the ternary mixture was determined by calculation of effective concentration values and calculation of combination index with polygonal analysis. The results show that single oligopeptides induce a distinct decrease of rotifer swimming speed in a concentration-dependent manner. The highest inhibition of the parameter was found in the animals exposed to anabenopeptin A and microcystin LR, both at 2,500 micrograms per liter. The animals exposed to the binary mixtures also showed the inhibition of the studied parameter. The highest decrease of swimming speed was found in the rotifers exposed to the mixture of microglycan-FR with anabenopeptin A and anabenopeptin A with microcystin LR at 500 and 500 micrograms per liter. Also, animals exposed to the ternary mixture showed the decreased parameter. The highest depression was found in the rotifers exposed to the mixture in which microgynin FR and microcystin LR was used at the highest concentrations. The analysis of interactive toxicity showed that the mixture of microgynin FR and anabenopeptin A induced slight synergistic effects. Also, slight synergistic effects were found in the mixture of anabenopeptin A with microcystin LR. Near additive effects were found in the mixture of microgynin FR and microcystin LR. Slight synergistic effects were found in the animals exposed to the ternary mixture as CI value was below 1. We found that neurotransmitter activity was depressed by single oligopeptides in a concentration-dependent manner. The highest decrease of the activity was found in animals exposed to 2,500 micrograms per liter of anabenopeptin A. The exemplary images show strong fluorescence intensity of the control animals, mostly in coronal region, mastax, yaw gland, and intestine region. Lower fluorescence intensity was noted in the animals exposed to the highest concentration of the oligopeptides. Binary and ternary mixtures of the tested oligopeptides also decreased the measured parameter. The exemplary images show that the animals exposed to the highest concentrations of the oligopeptides in the binary and ternary mixture showed much lower fluorescence intensity in comparison to the animals which were kept in the control group. Although the mixtures had inhibitory potential, the analysis of the interactive toxicity showed antagonistic and additive effects of the mixtures on rotifer catecholaminergic neurotransmitter activity. Antagonism was found in the mixture of microgynin FR with anabenopeptin A. Slight antagonistic effects were found in the mixture of anabenopeptin A with microcystin LR, and antagonistic effects were found in the mixture of microgynin FR and microcystin LR. However, near additive effects were found in the animals exposed to the ternary mixture of the oligopeptides. Our study also showed that fluorescence intensity of stained F-actin was decreased by single cyanobacterial oligopeptides in concentration-dependent manner. The highest decrease was found in animals exposed to the highest concentration of microgynin FR, 2,500 micrograms per liter, and the exemplary images showed that the animals exposed to the highest concentration of single oligopeptides manifested much lower fluorescence intensity when compared to the animals from the control group. The animals exposed to the binary and ternary mixtures of the studied oligopeptides showed inhibited fluorescence intensity of stained F-actin, and the exemplary images of the rotifers show the inhibition of fluorescence signal intensity when compared to the control animals. We found synergistic and nearly additive effects of cyanobacterial oligopeptides on F-actin fluorescence. Nearly additive effects were found in the animals exposed to the mixture of microgynin FR and anabenopeptin A. Very strong synergism was found in the mixture of anabenopeptin A with microcystin LR. Strong synergism was found also in the mixture of microgynin FR and microcystin LR. And strong synergism was also found in the animals exposed to the ternary mixture of the oligopeptides. As we can see, the CI value is 0.3 tenths. The analysis of muscle filaments showed that apart from the decreased fluorescence intensity, the exposed animals showed alterations of filament structure and changes, such as condensation of actin and filament breakages. Our study also showed that another invertebrate species, a crustacean, Daphnia magna, exposed to single metabolites manifested cytotoxic changes clearly visible as a red fluorescent color of propidium iodide in the transparent body. The highest level of cytotoxicity was noted in the animals exposed to the highest concentration of Syndospermopsin. Some level of cytotoxicity also expressed crustaceans exposed to microgynin. We also found that Daphnes exposed to the mixtures of cyanobacterial metabolites also showed increased red fluorescence, suggesting cytotoxic changes in their internal organs. On the basis of the obtained results, we can draw some conclusions. Firstly, single oligopeptides, microgynin FR, anabenopeptin A, microcystin LR, their binary and ternary mixtures induce toxic effects on the swimming behavior of Brachionus calyciflorus. Behavioral toxicity of cyanopeptides may be associated with decreased neurotransmitter activity and muscle F-actin degradation. In natural scenarios, various interactions of cyanopeptides in mixtures may be present, and total toxic effects may depend on the composition and concentration of the oligopeptides. Cyanopeptides induce cytotoxic changes in Daphnia magna. And the last conclusion, fluorescence microscopy was found to be a useful tool in invertebrate ecotoxicology. And the last word about our sponsor, National Science Center in Poland.